Okay, good morning everyone, good morning. Okay, let's wait for a minute, let others also join, then we'll start. Good morning everyone, good morning, good morning. Okay. Okay, so no problem, let others join. Good morning Shivani, good morning. Good morning Archana. Okay, so from today we will be starting what? The concept of enzymes. Okay. As I told you in the yesterday class, friends, from the topic of enzymes, we people has to read each and everything in the enzymes. Starting from most importantly the characteristic features of enzymes. What, friends? Starting from characteristic features of enzymes. Most importantly, friends, the types, then kinetics, complete kinetics we have to study. Okay, then followed by the inhibition. Okay, so while studying the kinetics, give the highest importance to the Michaels-Menten equation while studying the kinetics. Okay, good morning. At the same time, I want you people to understand, just understand AD-Hofstee diagram. Okay, give the highest importance to the Michaels-Menten, but try to study the other also. Okay, now let's start, friends. Many times we got the question from the characteristic features only in case of the 4 markers with respect to the matching question. So that is the reason what I am concluding is in the concept of enzymes, I want every one of you to study every single sub concept. Don't leave anything in the enzymes. Understand? Don't leave anything in the enzymes. Please try to study each and everything in the enzymes. Okay, now friends, let's study. Let's try to understand very clearly how can we define the word enzymes. Everyone follow me correctly. Try to understand every single point. So friends, right, enzymes are chemically what? Undoubtedly, friends, enzymes are chemically protein in nature. Now you all people has to get one valid doubt. What is the valid doubt that you people has to get? Does the chemical reactions will occur without enzymes or not? That is the valid question. Every one of you keep it in your mind, friends. Understand my points. Of course, the chemical reactions will occur without enzymes. There is no doubt about it. But the enzyme, that reactions cannot be an accurate reaction, and that reactions occur extremely slower. What, friends? That reactions occur extremely slower and that reactions are not accurate. So good morning. So every one of you try to understand my point very clearly, friends. Enzymes or the proteins, good morning, they are actually, friends, increasing the rate of reaction. How they will increase the rate of reaction, friends? Obviously by lowering the energy of activation. Anyway, friends, I will explain you what is this energy of activation. But in one line, I am explaining you, friends. Energy of activation means the amount of energy required to start the reaction. What, friends? How much amount of the energy is required in order to start that particular reaction? That is only, friends, we will be calling it as energy of activation. Understand my words correctly, everyone. Please follow me for 30-40 minutes continuously without disturbance. Please continue, understand every single point. The start of the reaction, or the amount of energy required to start the reaction, only we can call it as energy of activation. That energy of activation can be completely decreased by the presence of enzyme. Here comes one very important point. What is that? Enzyme function is what, friends? Converting the substrate into the product. That is the ultimate function of the enzyme. You all people know it. Now the question is that, do you think a substrate can influence the enzyme? Never ever. Do you think product can influence the enzyme? Never ever. So what I mean to say that the enzymes cannot be altered. Understand my words, friends. The enzymes cannot be altered, or the enzymes cannot be consumed during a particular reaction. So that's what we can say, that enzymes can be reusable. Please don't get confused. What, friends? Enzymes can be reusable because they cannot be affected by the substrate, they cannot be affected by the products. Please don't get confused with this particular point. Okay. Look into the picture, friends. As I told to all of you that enzymes are proteinaceous in nature. And we all people know very well, if the proteins wants to work very effectively, friends, what we generally require, friends, we will be generally requiring optimum temperature, and we generally requiring the optimum pH. We all people know it very well. Suppose example, if the temperature is more than the optimum, if the pH is more than the optimum, do you think enzymes will become extremely active, friends? No way. Correct, na? The reason is what, friends? The biological systems are very sensitive to the temperature changes. Whenever there is a fluctuation in the temperature, obviously, friends, enzymes also can lose their activity. What's the ultimate goal of the enzyme, friends? Remember, every student, enzymes are ultimately increasing the rate of reaction. How it is increasing the rate of reaction, friends? Without increasing the temperature. Because if the temperature increases, enzyme undergoes what? Enzyme undergo the process of denaturation. We all people know that. If the enzyme undergo the process of denaturation, how can it increase the rate of reaction? No way. So the enzymes work in such a way that, friends, enzymes always increase the rate of reaction without any impact or without increasing the temperature. How can they do this process? As I already told you, friends, ultimate goal of the enzyme is what? Lowering the activation energy. What is activation energy? The energy which is required to start a particular reaction. So because, friends, 37.2 trillion cells present in our body. Every student try to understand. 37.2 trillion cells present in our body. If they want to do a particular function, they compulsory require the activation energy. Without activation energy, none of the enzyme is not going to show any kind of a action. So what the enzyme is going to do? Enzyme will decrease this particular energy. As a result, friends, what is happening? The reaction will occur at a high faster rate. Don't confuse with that. Okay. Just now I have given you a hint. What is the hint I have given to you? Friends, enzymes will do the reactions in every step. Correct? Now, every step enzyme will perform the reactions. Enzyme play an important role in the metabolism, diagnosis, as well as the therapeutics also. Suppose example, you all people remember in the HIV analysis, what kind of test we will do, friends? We will do the test called as ELISA. Now you know, right? The ELISA is completely depending on what, friends? The ELISA is completely depending on the enzyme only. Correct? Now it's a kind of a enzyme linked test only. Because, friends, whatever the cell it may be, doesn't matter. Inside the cell, most of the biochemical reactions are linked to what, friends? Linked to the enzymes only. See, not only in the HIV analysis, friends, suppose example, if you take a myocardial infarction, which is a heart disease. Even, friends, myocardial infarction also we can understand by the study of level of a particular enzyme. So What I mean to say that enzymes plays a key role in the diagnosis also, enzymes plays a key role in the therapeutics as well. And friends, from this particular slide, I want every one of you pay attention from this particular slide. It's a very, very important slides. From now onwards, I want you to know the entire information. See the first point, because friends, characteristic features are very important. Enzymes are catalyzing only thermodynamically possible reactions. Friends, remember in the chapter of biochemistry in our CSIR exam, wherever you heard the thermodynamical properties, I want every student to read entire thermodynamical properties without leaving it. So what is thermodynamically possible reactions? You can simply remember, friends, the reactions which always follow the equilibrium, or are the reactions which always follow the Gibbs free energy. These reactions can be completely catalyzed or all the time catalyzed by this particular enzymes. And look into the second point, friends. Enzymes are not used or changed during the reaction. What do you mean by that word, friends? Which means enzyme cannot influenced by the reactant, enzyme cannot influenced by the product. Don't get confused. Even though reactant concentration is more, they cannot show any kind of activity in the enzyme. Even though product concentration is more, they cannot show any impact on the enzymes. I just now used a word, right? It catalyzes only thermodynamically possible, which means which maintaining the equilibrium. All the students look into the picture very carefully. They never ever change. They never ever change the position of the equilibrium and direction of that particular reaction. Please don't get confused with that, which means it never change the concentration of substrate or reactants. It never change the concentration of the products. It always try to maintain a equilibrium condition. Friends, I will tell you one very important line. Please remember this, every one of you. How generally enzymes are acting, friends? Enzymes are generally acting, friends, by forming a complex called as a transient complex. All the students remember, enzyme combined with the substrate, remember everyone, and it is forming what, friends? Enzyme substrate complex. Yes or no? Now every one of you try to understand my words very correctly, friends. Now this particular enzyme substrate complex, every student remember, this enzyme substrate complex is going to give rise to what? Enzyme plus product, is it? Where at a particular state, that state only we can call it as a transient state. I want all of you understand the point correctly. What is happening in the transient state? Everyone remember, either making the bond or breaking the bond is happening. What is happening, friends? Either making the bond or breaking of the bond is happening. By that the substrate can be converted into product. Wherever these reactions are taking place, that particular state only we can call it as a transient state. Don't get confused with this particular point. In the upcoming slides, I will little more clearly explain you about the transient state. And we all people know that, suppose this is the enzyme, is it? So you all know very well that a particular substrate whichever has to associated with the enzyme, it never associated randomly, is it, friends? Every enzyme by default, every enzyme by nature, they are having a particular binding site. The site which looks like a pocket, the site which look like a cleft, we generally call it as what site, friends? Active site. To this site, who will come and attaches, friends? The substrate or substrates will come and attaches. Now you know, most of the students will get confused, enzymes are what? Generally proteins. So what most of the students will think that enzymes are proteins, so they are very large molecules. There is a no doubt. Proteins are extremely large molecules, there is a no doubt. But what is the unique statement you have to remember? The active site is a very small. Friends, try to understand my words correctly. Protein is a large enzyme, which is a protein, which is a very larger molecule. All the students look into the diagram carefully. This is the enzyme, correct na? You only try to understand. The enzyme is a very large protein, but the active site is, you can see friends, active site is extremely smaller. Because why the active site is smaller, friends? Whatever the substrate or substrates that are attaching, they are smaller actually. They are smaller, friends. That is the reason you have to understand that active site is also smaller, and the moment the substrate will come and attaches to the active site, what is happening, friends? Active site undergo the structural change. You can also call it as what change, friends? Conformational change. The enzyme undergo the structural change or conformational change. Finally, what the enzyme is doing, friends? Enzyme is breaking down the substrate into product. Once again, I'll tell you in the upcoming slides how it can convert the substrate into product, friends. Remember this line without fail. Either making the bond or breaking the bond. This is the simple mechanism of an enzyme, friends. An example, friends, you take an example, sucrase enzyme. What is the function of sucrase enzyme? Break down the sucrose, yes or no? So what it has done? It break a particular bond. What is that particular bond? Glycosidic bond. We know that very well. By breaking the glycosidic bond, sucrase break down the sucrose into glucose as well as the sucrose. We know that, glucose and fructose. We know it very well. So same way, either in the making also, it is forming some particular bond. So friends, the state wherever making or breaking is happening, friends, that particular state only we can call it as transient state. Friends, please do not get confused with this particular point at all. Okay, now this is also extremely important slide, friends. Follow me correctly. Specificity. What is this particular specificity? Remember my words. Once again, this is the enzyme and this is the active site. Now the question is that how exactly a substrate will come and associated with the active site? That is the question. Remember my words. How exactly the substrate comes and associated with the active site? That is explained by a specificity. I will give the examples and I want every student has to remember compulsory examples. Specificity are of three types: absolute specificity, relative specificity, stereo specificity. Friends, one more very interesting point. What most of the students will think that to the active site, substrate will come and attaches very tightly. Once again, friends, this statement is also a very wrong statement. You people has to remember, in most of the cases, friends, the substrate associated with the active site through the multiple weak interaction. This is a very important question you have to remember. In a given 4-marker question, we can get, with respect to the statements. Always it is the multiple weak interaction. Friends, since the interaction is weak, that is the reason we can easily separate the enzyme and substrate. Because the interactions are non-covalent bonds. And another important statement every one of you has to remember compulsory is that how many amino acids will come close together and form this particular active site. As I already told to all of you, friends, active site is a very small. Active site is very small. Friends, this small active site is actually made up of 3 to 5 amino acids only. That's all. Only 3 to 5 amino acids come close together, friends, and they are forming this particular active site. That is the reason, friends, the larger substrates cannot fit into the every enzyme. Don't get confused with that. Okay. Now let me explain you, friends, the types of specificity. you people has to remember compulsory for the exam point of view. What? And it is very easy to remember also, friends. Absolute specificity. What is the point you have to remember about the absolute specificity? Name itself indicating na absolutely it will catalyzes only one. I can simply say that, remember my words, friends. In case of a absolute specificity, it is a catalyzing only one type of reaction. That's the unique point I want you to remember. It is catalyzing only one type of reaction for a single substrate. So let me tell you a very good example. Let's take an enzyme urease. Let's take an enzyme sucrase. Let's take an enzyme lactase. Now you only tell me lactase act on what? Lactase act on only lactose. Sucrase act on what? Sucrase act on only sucrose. Urease act on what? Urea only. Now you tell me, friends, does the lactase act on sucrase and urea? No. Does the sucrase act on urea and lactose? No. So what is the point you have to remember here, friends? In case of a absolute specificity, friends, there will be only one type of reaction will be catalyzed. The most important, it is specific for the single substrate molecule only. So urease catalyzes only the breakdown of the urea. Friends, this is called as what, friends? Absolute specificity. Do not get confused with this particular point. Next one, friends, all the students follow my words correctly. The next one is group specificity. Friends, what you have to understand by the word group specificity. Remember, here also it is catalyzing one type of reaction, which means that it is catalyzing the reaction with respect to the similar substrate. Let me explain you elaboratively. It is catalyzing the reaction with respect to the similar substrate. What do you mean by that? Friends, let me take you one very good example of a enzyme. Hexokinase enzyme. We all people know very well, friends, what is the function of a kinase enzyme. We all know very well. The ultimate function of the kinase enzyme is addition of a phosphate group. Yes or no? Of course, yes. Addition of a phosphate group. And where it is adding the phosphate group? It is adding the phosphate group only to a group of sugars. What do we call it as? Hexoses. Now all the students understand my words very correctly, friends. Do you think hexokinase will go and add the sugars, add the phosphate group to the pentoses? Never ever. It never add. Why? The reason is very simple. The hexokinase is catalyzing only one type of reaction, and most importantly, friends, it is catalyzing the reaction only to a similar substrate. So hexokinase act on only hexoses. It never act on any other group. So that is called as what? Group specificity. Friends, please don't get confused. Okay. What about the next one, friends? Next one we can call linkage specificity. Friends, the moment you heard the name of a linkage specificity, the common point has to strike in your mind is bond. The common point has to strike in your mind is bond, which means in the linkage specificity also there will be a catalyzation of only one type of reaction with a specific to a type of bond. Let me give you some more examples here. Let's take an example of a enzyme chymotrypsin. Right. So let's take an example of a lipase enzyme. Right. Okay. Friends, let's take an example of an enzyme glycosidase. All the students try to understand what I am talking about. Friends, what is the function of a chymotrypsin? It is the one only cleaving which bond, friends? Peptide bond. You all people know that very well. And friends, what's the function of a lipase, friends? Lipase is the enzyme which will only cleave the ester bond. And friends, what about the glycosidase enzyme, friends? it can only cleave what bond, friends? It can only cleave the glycosidic bond. So you tell me, do you think a chymotrypsin can break the ester bond? Never ever. Do you think chymotrypsin can break down the glycosidic bond? Never ever. Do you think a glycosidase enzyme will cleave the peptide bond? Never ever. So what's the point I want to conclude here? I want to conclude a point that in case of a linkage specificity, there is only one type of reaction, friends, and that too with respect to the specific type of a bond. That is the point I want you people to remember. I don't know what you do. I hope everyone understand this concept very correctly, and I am requesting all the students remember the examples, because in the exam point of view, especially in the matching section, they will give the examples and they will ask us identify what kind of specificity it is. What, friends? They'll give the example and they will ask us identify what kind of specificity it is. Okay. Now all the students follow me. Friends, understanding the characteristic features are very important. If you don't understand the characteristic feature, friends, you can't understand the kinetics at all. So always try to remember all the, characteristic features correctly. So this is the substrate. Now every student has to tell me the difference between the substrate and reactant. See, actually, friends, originally we use the word reactant. Whenever, remember my words, friends, whenever a reactant participated in the biochemical reaction, whenever a reactant participated in the biochemical reaction, then we can call it as the substrate. So what is the point you have to remember? Both the reactant and substrates are same only. Depending on the reactions that are involving, we are calling with a different names. Every student pay attention. The reactant in biochemical reaction, that is what generally we termed as what, friends? Substrate. Please don't get confused with that particular point. Now remember, when a substrate bind to enzymes, follow me, friends. When a substrate bind to an enzyme, it forms an enzyme substrate complex. All the students look into the point very clearly. Once again, I am reminding you an important statement. Here, when the substrate will come and join with the enzyme, friends, here obviously what kind of interactions will be formed, friends? Here the type of interactions will be occurring always, friends, non-covalent interactions. They are not a greatly stronger interactions. Okay. Listen, friends, I'm going little faster because this concept is not a difficult concept, right? When we are studying the concept of kinetics and inhibition, I'll go little slow and I'll explain you very depth. Now remember, friends, in order to explain how a particular substrate will come and associated with the enzyme, there are multiple models were proposed. What, friends? There are multiple models were proposed in order to explain how the substrate and enzymes will come and associated with each other. What are those, friends? Every one of you try to understand my points correctly. Understand. There is a lock and key model. There is a induced fit model. Is it or not, friends? Of course, yes. Now, every one of you remember my words. Why the lock and key model is not widely applicable. All of you keep it in your mind. Lock and key model is a old model and not applicable. It is not applicable to all enzymes. Now the second point, induced fit model. This is a new model, and understand, friends, it is the one applicable to all enzymes. Now I will explain you why lock and key model is not applicable, why only induced fit model is applicable for all the enzymes. Let me tell you in a simple way. I want you people to remember few unique statements. In case of a lock and key model, every student follow my words correctly. In case of a lock and key model, this is what site? Active site. Every student keep this point in your mind. The active site is extremely rigid in shape. What friends? The active site is extremely rigid in shape. So what? If it is in rigid in shape, what is going to happen? Whatever understand my words correctly. Since the active site is rigid, friends, whatever the substrate exactly fit here, exactly fit into this particular active site, only that can convert it into product. I'm sorry, only that substrate can be converted into product. Suppose example, all the students follow my words correctly. What is the shape of the active site? This is the shape of the active site. And here what is the shape of the substrate? Substrate is also exactly looking the shape of the active site. Then this particular substrate can easily fit. Suppose example friends, the substrate shape is like this. The substrate shape is like this and the active site shape is like this. Do you think a substrate can fit, friends? There is a no way. Substrate never ever fit into this particular active site. So what's my ultimate goal, ultimate conclusion here? In case of a lock and key model, the shape is rigid. So whatever the substrate with the matching shape, whatever the substrate with matching shape that can fit, friends, it is exactly like a lock and key. Now you tell me, friends, if I want to open a specific lock, I need a specific key, yes or no? If I want to open a specific lock, obviously I require a specific key. I can't open the lock randomly, yes or no, friends? I can't open the lock randomly. Similarly, friends, what is the point you people has to understand here? The substrate with the matching shape only can fit here. That is the reason we are calling it as the lock and key model. Not at all widely used, not at all applicable, not at all work for all the enzymes. Now, what is the second one, friends? Second one we can call it as, good morning, good morning. Second one, friends, we will be calling it as the induced fit model. What's the unique point about the induced fit model? All the students try to understand what exactly I'm saying. Here I told every students, the moment you heard the name of a active site, I told to all of you friends, remember it is rigid in shape. This is the point I told to all of you. Similarly, friends, the moment you heard the name of an active site, you people has to remember here one common point. Here it is the flexible. Which is flexible? The active site is flexible. All the students look into the diagram. This is the shape of the active site. Remember, friends, don't get confused. Good morning, good morning. This is the shape of active site. And this is the shape of substrate. This is the shape of the substrate. But what is the interesting point? What is the interesting point? The moment this substrate will come and attaches, you know what is happening, friends? This active site will exactly change the structure like this. So what you have to remember my words, you have to remember my words that here active site is flexible. It can changes, understand my words, it can changes according to the shape of the substrate. It changes according to the shape of the substrate. It doesn't matter to us. Very good. It undergo the conformational or structural changes due to its flexibility, and it can exactly fit into the shape of this particular substrate. That is the reason, friends, all the enzymes can use as induced fit model when compared to the lock and key model. Don't get confused with this particular point. Okay, friends, these are all extreme basic. I will tell to all of you what you people has to remember for the exam point of view, especially for the 4 marker. Now remember, when the enzyme substrate complex is going to be formed, friends, this is unique one. Please every student pay attention without having confusion. This is the enzyme combined with the substrate, and what do we got here? We got enzyme substrate complex. Now you tell me, tell my word, see, understand my words. Our goal is to form the enzyme substrate complex? No, our goal is to form the product. This is the area where most of the students are confusing. When the enzyme and substrate come close together, there is a formation of enzyme substrate complex. Every student remember my words. Here, whatever the interactions that are formed, friends, these interactions only we can call it as non-covalent interactions. What do we call it, these interactions, friends? We call it these interactions as the non-covalent. But friends, this is the statement I want every student has to remember. This is what state? Transition state. I have clearly explained it to all of you. Friends, transition state is the state where making of the bond or breaking of the bond is taking place. Now here, what kind of interactions that we can observe? Friends, here the type of interactions are covalent interactions. We will break that particular covalent bond, or we will make that particular bond, and we can make the product. Once again, friends, please do not get confused with the concept. When the enzyme and substrate combine with each other, when we are forming a enzyme substrate complex, the interactions are non-covalent. But friends, when the substrate is converting into product, there is only two ways of converting the substrate into product. What are the two ways, friends? Either by making the bond or by breaking the bond. During this time, the interaction will be the covalent, because in a given 4-marker question, the statements always make you confuse. When the covalent bond breaking or making occur, keep it in your mind only in the transition state before forming a product. Okay, now all the students look into the picture. The moment product got released, what is happening? Tell me, friends. The enzyme once again undergo the structural change, or the enzyme undergo the conformational change. Understand my words. If the enzyme undergo the conformational change, do you think, friends, do you think enzyme can associated with the product? Never ever. Enzyme never associated with the product. It is ready for the another substrate attachment. You people has to remember my words correctly. Enzyme and product always having the lower affinity. Always having the lower affinity. Enzyme and substrate always having the higher affinity. Always remember this particular statement. Friends, the moment product formed, what is going to happen, friends? Immediately this enzyme will undergo the conformational changes. It is getting ready for the another to attaches with the another substrate. Okay. So far till this particular slide, whatever we have discussed, it is very easy part. It's not a difficult part, and we may or may not expect the questions. But from now, this particular slide onwards, all are extremely important. Great chances are there that we can expect the question, and we got the part C questions, we got the part B questions from this particular concept. We got in the matching also, we got in the statements also, we got a direct part B questions also. So I am requesting all of you try to understand the concept correctly, and most importantly, these are all the concepts which you have to remember. Okay, when it comes to the kinetics, except formulas, you don't need to remember, everything else to be understood very elaboratively. But here you have to mostly understand the concept. Okay, let's follow now. Remember everyone, the enzyme, see friends, I have defined one very clear-cut definition. What is that? enzymes are chemically proteinaceous in nature. You know what, most of the students will get confused with this particular statement. When we use a statement that enzymes are protein in nature, what people will think that 100% of the enzyme is actually protein only. That is not the true statement. What I can say that, friends, let's consider 80 to 90% of the enzyme is protein. 80 to 90% of the enzyme is protein only, friends. What about remaining 10 to 20% of the enzyme, friends? That is the non-protein. Friends, one more valid doubt every student has to get. What is the valid doubt you people has to get for the exam? Sir, this non-protein part is only 10 to 20%. Now my question is, if the non-protein part is absent because it is consists of a very less percentage, do you think protein part of the enzyme will be active? I can tell you, friends, it never ever be active. In one word, I want you to remember that without a non-protein part, the active site of the enzyme never ever be active. The point every student has to remember is that non-protein part is the one which is actually forming the active site. What, friends? Non-protein part is the one which is actually forming the active site of the enzyme. So my friends, without non-protein there is no way of formation of the active site. Okay. Enzyme having a protein part, having a non-protein part. Every student remember, what do we call this protein part? We can call the protein part as apoenzyme and always inactive. What is this particular non-protein part? We can call the cofactor. Is said to be the non-protein part, and the cofactor only forming what site? Active site. Now all the students look into the picture very correctly. This cofactor come and attaches to what, friends? Come and attaches to the protein part. Then what it forms? Look into the picture. It is forming the active site. The moment it forms the active site, then only substrate will come and attaches. So what I want you people to remember, apoenzyme plus cofactor. Together, what do we call it, friends? Together we can call it holoenzyme. Friends, what is the full form of holo? Holo means complete. Holo means complete. When the enzyme becoming complete, when there is a both protein part and non-protein part are present, then only it will become complete. Otherwise, friends, it is never be completed. Okay, just give me one moment. Just 1 minute. One moment. Okay, now listen. So what I want you people to remember for the exam point of view is that, friends, remember my words. The cofactor, that is the prosthetic group, and coenzyme. Friends, right now whatever the properties that I am going to explain to all of you, remember the properties as it is, and most importantly for the exam point of view, remember the examples. All the students look into this. Non-protein part is called as apoenzyme. Protein part is called apoenzyme and non-protein part is called as cofactor. Friends, every student please pay attention. This particular cofactor or the non-protein part is of two types. What is that? One we can call the prosthetic group, friends. Another one we can call the coenzyme. Whatever the statements that I am telling you right now, please remember the statements as it is. The moment you heard the name of a prosthetic group, always remember it is an inorganic molecule. Every time. You all know that inorganic molecule means it is always small molecule. The moment you heard the name of a coenzyme, Always keep this point in your mind. It is a large organic molecule. And friends, what is the point you have to remember? Since it is a large molecule, friends, since it is a large molecule, they associated with the protein part very loosely, because it can't fit into the protein part very easily. Don't get confused. It is loosely bound to the apoenzyme. What is the reason? The reason is it is very large in size. But friends, if you look into the prosthetic group, there is a small inorganic molecule or atom. Remember friends, since it's a very small molecule, right friends, the attachment is extremely tight attachment. I don't know what you do, friends. I want you to remember this compulsory. Okay. Look into the picture. Now we have to study see what is the non-protein part. Non-protein part is the cofactor. There are of two types, coenzymes and prosthetic group. Coenzyme is loosely bounded. See my friends, a very important point. I have used a word that coenzyme is loosely bounding. It is an organic compound. Now what is the point every student has to understand? Since the attachment is a loose attachment, I can break it easily. Why? Because the interactions are non-covalent interactions. The best example which you have to remember for the exam point of view, vitamins are compounds derived from vitamins, right? Friends, when it comes to the prosthetic group, what every student has to remember, it is a non-protein part. It will be very tightly bound to the enzyme. My friends, once again the unique statement, I am using a word tightly bounded. Once again the bond will be covalent bond, because you have to remember these bonds also with respect to the statement questions and what we got in the exam. Okay, so this is the direct question we got in the exam from this particular table. Okay, just 1 minute friends, just 1 minute. Yeah. Now I want every one of you to understand here the cofactors. I know friends, this is really difficult concept to remember. Why it is difficult is you have to remember a lot. But twice we got the 4-marker question, twice, and that too in the matching only. I know it's difficult to remember all, so at least whatever I'm marking, I want you see. If you can remember the entire chart, it is really good. I really appreciate it. But if you can't remember all, remember the copper, iron, potassium, magnesium, and the most important one is the zinc. These 5 I want you people to remember. If not, try to remember each and everything. Let's take an example of cytochrome oxidase. What is the cytochrome oxidase, friends? Cytochrome oxidase is the most important enzyme that is used in the electron transport chain. Correct or not? Of course, yes. Correct, na? But what is the unique statement you have to remember? If the cytochrome oxidase enzyme wants to work effectively, friends, compulsory it requires the copper as their major cofactor. Even friends, now here comes another point. If cytochrome oxidase is using the copper also, cytochrome oxidase is using the Fe2 plus or Fe3 plus also. Now you will get a confusion that in the exam if they give both, which one we have to choose? Undoubtedly friends, if they give both for one option only, you can choose any. Undoubtedly they'll give the answer, but they never give that kind of options at all. If they give cytochrome oxidase Fe3 plus, they never give the copper. Suppose example, if they give this particular copper, they never ever give the Fe3 plus. Always remember that. But you keep the point in your mind. Cytochrome oxidase is using both Cu2 plus also as well as the Fe2 plus or Fe3 plus. And another important, catalase. Catalase is the enzyme. Catalase and peroxidase are the enzymes which are breaking down the an important free radical H2O2. If this particular enzyme wants to be active also, there will be a requirement of iron. Very important. And the most important enzyme in the glycolysis, which is involved in the 10th step of glycolysis, which is converting phosphoenol pyruvate to the pyruvate. What you have to remember, friends, there will be a utilization of what, friends? There will be a utilization of the potassium only. Don't get confused. Okay, now friends, another important one, magnesium. Hexokinase enzyme, glucose 6-phosphatase enzyme, pyruvate kinase enzyme. Where all these three enzymes are present? All these three enzymes are present only in the glycolysis only. Okay, now so for the three enzymes there will be a requirement of magnesium. And very important is the zinc, which was asked in the exam. Carbonic anhydrase enzyme, alcohol dehydrogenase enzyme, carboxypeptidase enzyme. What is carboxypeptidase enzyme? Which is actually cleaving the carboxy terminal domain of the peptide. The carboxy terminal domain of the peptide can be break down by this particular carboxypeptidase enzyme. And this enzyme only uses zinc as their major cofactors. I don't know, friends, what you do. You have to remember the chart. Keep it in your mind. Whichever we neglect, the questions will come from there only. So wherever you see the charts or wherever you see the tables, I am requesting every student read, especially in the chapter like biochemistry, especially in the chapter like ecology, especially in the chapter like unit 13 techniques. Please don't neglect this at all. So friends, now remember the point which we have to remember for exam. What are coenzymes? In one line, how can I define the word coenzyme? They are the one group transfer reagents. They can transfer hydrogen, they can transfer electron, they can transfer different type of atoms depending on the condition. Where you can expect the questions from the examples. Everyone remember, here you no need to use the brain, friends. Lot of information to be remembered. Some of the enzymes are metabolite coenzymes. Some of the enzymes are vitamin derived coenzyme. Both are important only. Friends, one interesting point is what? Vitamins, how much concentration they will be required in our body? We all people know that vitamins are required in our body extremely low concentration. Very low concentration. The enzymes, vitamins will be required. But what is the point? Even though they are required in the low concentration, the deficiency of that low concentration can cause the diseases. So the point you have to remember, even though vitamins required in the low concentration, they are having a significant importance in our body, because most of the enzymes depending on the vitamins. Why? Most of the enzymes, vitamins are acting as a coenzyme for that particular enzyme to active. Look into the picture. Here I have given you two type of classification. One is metabolite enzyme, one is vitamin derived enzyme. Every student has to remember both. Let me tell you the examples of metabolite coenzymes. Remember all the examples without fail. ATP, S-adenosyl methionine. You know, right? S-adenosyl methionine. We have studied in the molecular biology. Shortly called as S-adomet. Third one, 5, 6, 7, 8 tetrahydrobiopterin. These three are the examples of metabolite coenzymes. I really don't know what you do, but I want you people to remember them compulsory. Let's talk about the ATP. What is the function of ATP? We know that none of the cell never be functional without the energy. So energy is coming from what group? This phosphate group. So who is the donor of that phosphate group? ATP. We people especially studied this iodomet in the 5' capping. What is the function of this particular 5' friends? Adenosyl methionine. This is the one which is acting as a donor of the methyl group. Right now, you know, right friends, to whom it is donating the methyl group? It is donating the methyl group to the 5' capping. Correct na? That's why we can call it methyl 7 guanine. Yes or no? Because the methyl group is donated to the 7th position of the guanine, which we studied in the molecular biology. That's why we are calling it as the methyl 7 guanine. Don't get confused. Then 5, 6, 7, 8 tetrahydrobiopterin. What is its function? It is acting as a cofactor for nitric oxide synthase enzyme. Friends, what is the function of a nitric oxide synthase? It is the enzyme which is converting arginine into citrulline. When the arginine can be converted into citrulline, nitric oxide can be released as a end product. Nitric oxide can be released as a byproduct, and the enzyme that is catalyzing the reaction only we can call it as a nitric oxide synthase enzyme. Please don't get confused with this particular point at all. So these three are what, friends? Metabolite derived coenzymes. You have to remember them. Then what are the vitamin derived coenzymes? NAD, NADP plus, FAD and FMN, TPP, PLP. The remaining thing we will discuss in the 11:30 AM class. Okay, it's a very broad concept. But today only in the 11:30 AM class and in the 3:30 PM class, at least we have to finish till the kinetics. By that we can finish the inhibition in the tomorrow morning class. Okay, is it clear everyone? Please let me know everyone, is it clear? We'll meet in the 11:30 AM sharp in the app. Anyone having any kind of a doubt, please let me know. Yeah, someone asked a doubt. What is that? Okay. Deepika, listen to my words. There is an enzyme called, Deepika, listen. There is an enzyme called as creatine kinase. There is an enzyme called as creatine kinase. Okay. If this enzyme concentration is more, which indicates that the particular person having the myocardial infraction. You are understanding my words? So the excess quantity of this enzyme only helping us to identify whether the person is having the myocardial infraction or not. Actually, this creatine kinase is present in all the muscle tissues. Remember, when this creatine kinase is releasing, actually whenever a myocyte got injured. Understand my words. Whenever a myocyte or a muscle cell got injured, especially myocardial tissue of the heart got injured, this enzyme concentration is increases. If the enzyme concentration is increases, which means that there is a myocardial infraction occur. That is the point I want you to remember. Got it? Okay. Okay friends, I'll see you at 11:30 AM sharp in the